Hi everyone. I'm excited to be involved in the Ridge to Reef Summer Institute this year, and I'm going to give you a little insight into the research that I do and a tutorial on time series analysis. So because I'm talking about time series analysis in my tutorial, I decided to present some of the work that I've been doing over the last couple years using plankton time series data from many different countries to ask questions about how the environment shapes interactions and stability in food webs. So before I get started, I thought I'd start with the acknowledgements because this project originated during an opportunity for um, early career scientists, both soon-to-be and recent graduate students who work in aquatic ecosystems. So I think there's probably many of you in Ridge to Reef who will be eligible for this program, and I wanted to draw your attention to it. This is called Eco Days, and it's a symposium for collaborative interdisciplinary aquatic research, and you get to join forces with about 30 other of your peers to write collaborative papers. And it really is one of the best meetings and programs that I've been involved in. So if you work in aquatic systems at all, I suggest that you look into it. I also wanted to call out Tanya Rogers, who is my close collaborator on all things time series. She's at NOAA in Santa Cruz now, and she has contributed greatly to all of that I will present today. So jumping into it, part of my talk today will be addressing one of the oldest questions in ecology. What controls the distribution of biomass within and among ecosystems? There's some areas in the world, like the forests, that contain a ton of plant life, and others, like the desert, that contain less. There's also a lot of variation in the relative biomass of different trophic levels. So, for example, the plains of Africa support a very large biomass of animals relative to the plants that are there, compared to places like the temperate forest. So, what causes that variation across the globe has intrigued ecologists for centuries. So, narrowing in on one example, here in this photo, we have kelp and fishes and all the other marine life that you can't see. So what determines their relative abundances to each other? So at the base of the food web, we have the resources that are available in the ecosystem, which can determine the biomass of the higher trophic levels through bottom-up or resource control. So this is where the amount of primary productivity can determine how much biomass can accrue at higher trophic levels. In about the 70s, there was this new idea, which is that predators can also control biomass distribution in ecosystems through cascading effects through their prey all the way down to primary producers. So here I have this little seal who is exerting consumer or top-down control on its prey. The balance of these top-down and bottom-up pathways are important to understand because the relative importance of them likely depends on the environment. And as humans shift environmental conditions, not only are we directly influencing the survival of different species in these ecosystems, but we're also shifting the strength of species interactions and reshaping the entire food web. Now we know that both consumers and resources can have huge impacts on the environment. So I've just included two photos here of some iconic experiments, one manipulating consumers on the top and one manipulating resources on the bottom. So these are both photos of whole lake experiments where the lake was split in half with this impermeable curtain sent down the middle. And in the top, you can see that predators have this large impact on water clarity, where impacts of bass trickle down through planktivorous fish, herbivorous zooplankton, down to phytoplankton or algae. And on the bottom, you can see that adding phosphorus causes these algal blooms. So both of these are experiments where you don't need statistics. You can just look at them to see that there's this tremendous effect. But although these are iconic examples about how important consumers and resources can be in these systems, they don't tell the whole story. So there's also been other manipulations of whole lakes or ponds that have had much, much weaker effects or even no effects at all. So how can that be? Well, So there's a lot of different hypotheses about when consumers and resources are going to impact whole ecosystems, but one explanation is that the environmental conditions in these locations determine the strength of species interactions. Bottom-up and top-down species interactions in food webs are variable over space and time and can differ with a variety of abiotic or biotic conditions. So one important example is that with warming temperatures, there is the prediction that top-down or consumer control will increase because metabolism is more temperature sensitive than photosynthesis. Species traits could also influence these interactions. So for example, here if phytoplankton, they can become more or less palatable through spines or growing clonally, changing the top-down effect from zooplankton. And finally, species diversity at both of these trophic levels is expected to influence dynamics through complementarity. So how do we actually study how these species interactions vary? So a lot of our insights have come from experimental studies, such as these nutrient additions or predator removals that I showed earlier. But these experiments are often limited in spatial and temporal scope, and the manipulations tend to be rather extreme. So here they're removing fish or adding fish, whereas in nature it's more likely that food webs are going to be affected by variation in the abundance through time, not necessarily the wholesale addition or removal of a species, or trophic level in this case. Also, these experiments aren't always tractable, so we haven't done a ton of whole lake experimentation with removing and adding trophic levels because they're not always tractable. So what can we do? Can we make better use of observational or monitoring data to learn about species interactions? There's been some attempt at this, and most of the work has been looking at the change in two adjacent trophic levels, their abundance through time. So if they're positively correlated, that's been taken as a signal of the system being bottom-up controlled. So it's phytoplankton increase, zooplankton increase, that means that there's more energy for zooplankton, and that is an example of bottom-up control. Or if they negatively covary, so as zooplankton, say they increase in abundance and chlorophyll decreases, that would mean that it's a top-down effect. So they are, the zooplankton are controlling the abundance of their lower trophic level. So that's kind of been the approach so far, and what that does is it takes an ecosystem, it takes time series data from that ecosystem, and boils it down to one answer. So this is a bottom-up or top-down controlled ecosystem. But really, we know that these species interactions vary through time as well. So what we really want is an approach that does not assume interaction strengths are constant and allows them to vary through time. It also should recognize that both bottom-up and top-down effects occur simultaneously and provide information on whether they're both strong or both weak, rather than just the relative importance. So luckily we have some of those types of models now that we can use. And the work I'm going to be presenting today uses this empirical dynamic modeling approach, and which I'll refer to as EDM. And EDM is on the far end of the continuum of modeling approaches, from these mechanistic models where the relationship between each constituent pair of species is known and included explicitly in the model, to these equation-free methods that are more akin to a black box, where you don't know these constituent interactions between each individual component, but you can still model the dynamics of the ecosystem as a whole. And to do that in EDM, we use time series data and the information those time series contain to understand how a system behaves and how it will, or predict how it will behave in the future. So let me try to describe that a little bit more. So here is an example of time series data, but instead of being plotted by time, here I'm showing each point as the resource abundance. So in our examples, it will be phytoplankton abundance, and then the consumer growth rate on the y-axis. So here you might just think that these are positively correlated with a little bit of scatter around this point. And historically, we would think, okay, as resources increase, consumers increase, so that this system is bottom-up controlled. The amount of resources available set the limit on consumer growth. But with EDM, we think about how things change through time. So what I've added here is all the lines that connect these points. And you can see that through time, so these lines are representing time, you get this kind of figure 8 or butterfly shape. And these are actually the dynamics you get from this food chain model, if you this deterministic food chain model. And when you look at the relationship between resources abundance and consumer growth rate through time, you can see that it's not just this static positive relationship, but instead those relationships between these two things change. So at some points in the time cycle, we have a positive relationship where there's an increasing consumer growth rate with increasing resources, whereas other times there's no relationship or a negative relationship. And so what EDM does is it actually computes these relationships at each and every time point. And it does that by creating these locally weighted regressions. So instead of thinking about regressing or creating these models through time where you're making predictions based on what has happened most recently, instead you're looking at what is happening around the last time that your resources were at this level. So when you're in this region of resource abundance, what do we expect consumer growth rate to do? And we expect it to behave similarly as it did last time the resources were at that abundance. And similar to what I was describing before, the way that we get species interaction strength out of these kinds of models is looking at the slope coefficients. So again, the positive relationship would suggest bottom-up control, and this negative relationship could suggest a more top-down effect of consumers on resources. So using these methods, we set out to ask the question of how bottom-up and top-down species interaction strengths vary over time in natural systems. Again, using EDM allowed us to not only look at classify a system as bottom-up or top-down control as a whole, but actually look at how these things varied through time. And we were also interested in what environmental drivers were associated with changes in species interactions to think about how climate change might impact the relative importance of different interactions in food webs. And we also asked if the environmental drivers, differed across versus within sites. So I've definitely alluded to this, but we, as our study system, use time series data from lakes around the world. So many of them were in the United States. There's a bunch of lakes in the North Temperate Lakes LTER and down in Florida. And then a couple in Europe, one in Japan, and one in New Zealand. The issue with EDM is that it's very data intensive. So not only do we need a lot of time points, but we also can't have gaps in the time series data. So there's a lot more lakes that are sampled and have been sampled for, decades, but they're usually only sampled in the summer. And we required lakes that were sampled all year round. So these are the ones that have these monthly sampling points that we were able to to capture. These lakes vary in a lot of their environmental conditions. So for example, the nutrient conditions in these lakes were wildly different. We measure that with this thing called the trophic state index from oligotrophic up to eutrophic. And just looking at a couple of these photos, you can see from the New Zealand lake in the oligotrophic up to the Florida lake in the eutrophic section, you can see just from these Google images that it's very clear from the greenness which lake is highly eutrophic. So for these time series, we were interested in modeling the bottom-up effect of phytoplankton on herbivorous zooplankton and the top-down effect of zooplankton on phytoplankton. And to do this, we fit 2 models for each lake, one for each of these processes. And we did that using this our EDM package. So the bottom-up and top-down models look pretty similar, and I'll talk through both of them. So in the first model, this is the bottom-up model. Our response variable is zooplankton growth rate, and the predictors are the zooplankton abundance from 1 month ago and 2 months ago. So those are accounting for density dependence within trophic level effects on growth rate. We also have this seasonal predictor, so those are just some sine waves that account for seasonality that are not necessarily captured in our environmental variables. And we then have the cross-trophic level effects of chlorophyll 1 month ago and chlorophyll 2 months ago on zooplankton growth rate. And the effect that we'll be talking about from here on out is this effect of chlorophyll 1 month ago on zooplankton. So that's what we're calling the bottom-up effect. So basically, if that's positive, if increasing chlorophyll increases zooplankton abundance, that's suggesting that this bottom-up effect is positive and causing growth. Or you could have, as chlorophyll increases, zooplankton decreases. So you can have a bottom-up effect that is negative. So for example, if there are toxic phytoplankton in the assemblage, that could drive that negative effect. And we can capture both of those positive and negative relationships as a bottom-up effect in this model. In the second model, the predictors are the same, but now the response variable is chlorophyll growth rate. And the top-down effect is now the zooplankton abundance from a month ago and its effect on chlorophyll. So we can imagine that it is often negative, where if there's high abundance of zooplankton, they're grazing down the chlorophyll, and so you're going to have this negative effect. But you could also imagine this top-down effect could be positive if there's a lot of zooplankton, they could be recycling nutrients in the water column, causing the growth of more chlorophyll. So what essentially happens is that you start with a time series of both zooplankton and chlorophyll abundance, and you end up with a species interaction strength for both bottom-up and top-down effects. So these are the coefficients that we get. And in this lake, as you might expect, the bottom-up effects tend to be positive, so they are above 0 here. And again, that means that with more chlorophyll, you have more zooplankton. So we get that kind of expected positive impact, positive bottom-up effect. And then the top-down effects, in orange here, they tend to be negative or below 0. And that's when there's a lot of zooplankton in the past, it reduces the growth rate of phytoplankton. But the other thing that you can see is that there's a lot of variability. These are going up and down, and sometimes even crossing the 0 line. And you might, it looks a little bit like they mirror each other, which is true, but it's not something that mathematically had to happen with the way that we designed our models. It just emerged from these 2 separate models. So there seems to be something, and you'll see that in other models too, where the strength of both bottom-up and top-down control seem to be happening simultaneously. Okay, so now we have this continuous variable that tells us about the strength of species interactions, and now we're interested in what predicts the strength of those. So we looked at both seasonality, so these are the sine waves that were included in the model, and then some particular environmental variables. So we have the trophic state index, that gives us information about the nutrients in the lake. We have water temperature, and then species richness. We model this both across lakes, so we put in all the interaction strengths from all the lakes and their environmental variables to look at how those things scale across lakes. And then we're also interested in, within lakes, how the interaction strengths change. And for those, we center the predictors and scale them. And basically what that is doing is we're saying, in a lake, if we think temperature influences species interactions, well, it doesn't really matter what your mean temperature is necessarily. It might just matter if you are in a warm part of the year or a cold part of the year, or in a warm spell or hot or cold spell, things like that. So that's the within lake models. So what do we find? So across lakes, both bottom-up and top-down effects were driven by nutrients. And they explained a lot more variation than even the next closest predictors. Whereas within lakes, seasonality seemed to be the most important thing, mostly in the bottom-up effects. Although I will also say that these within lake models, we were never able to explain very much variation in interaction strength at all. So also note that in both cases, temperature and species richness, which we really did think, based on previous predictions, that there would be a strong effect on species interaction strengths, we did not find strong effects of those at all. In terms of what those relationships look like, so here's the interaction strengths plotted against trophic state index. And to reduce the clutter, these are just the means and standard deviations for each lake rather than individual time points. And we find this curved relationship where at mid trophic state index, you have positive bottom-up effects and negative top-down effects, which we might expect under these typical conditions that we've been talking about. But at really low nutrients, so these really oligotrophic lakes, we see that the top-down effects are actually positive. And again, that means that zooplankton are promoting rather than suppressing chlorophyll growth. So this is consistent with the idea that in these super low nutrient lakes, that zooplankton can actually excrete and recycle nutrients, promoting algal growth. In contrast, at these really high nutrient lakes, their species interactions are both closer to zero, and that seems consistent with the idea that inedible algae, cyanobacteria, toxic algae, they're often dominant source of chlorophyll in these lakes, and zooplankton do not have strong top-down control on those types of species. Okay, so next we have the effect of seasonality within lakes. So this is the interaction strengths within each lake plotted against month. And you can see that many of these show some seasonality, which we've already showed is not directly related to temperature or species richness or any of the other nutrient drivers that we measured. And the magnitude of both top-down and bottom-up effects appears to be the greatest in the warmer months. So that's months 4 through 8 in all of them except for Lake Taupo, which is in New Zealand. So the warmest months are in our winter, so 11 through 2. And the top-down and bottom-up effects being the strongest in the summer is consistent with this idea of a seasonal reset. So with these effects being strongest as lakes turn over and things emerge from the sediments and kind of are assembling this community, and then there's this decline in the strength of trophic coupling through time. So just thinking about the top-down effects, you can imagine that as Daphne are emerging from the egg bank and you're getting a lot more algal production, those Daphne are going to have a really strong top-down effect and graze the algae down to lower concentrations. But at some point, there's going to be pressure on those algae to become more inedible, and we see this in time series as well, where over time, throughout the summer, phytoplankton become more defended, more structure, more clonal, and there's a reduced top-down effect. And so it seems that what's happening within these lakes is this seasonal pulse where you have both bottom-up and top-down control increasing and then decreasing through time. So to conclude this part, we see that species interactions are clearly not static in time, and different drivers are associated with this variation, depending on whether we're looking within or between lakes. So between lakes, it seems like nutrient status really changes how these interactions affect your dynamics. And it's likely because when you're comparing a bunch of different lakes, there's a greater range of environmental variation captured when looking across lakes. When you're looking across lakes. When you're looking within lakes, there's not as much environmental variability, so the variability is smaller, and we did not see any real relationship between the environment and species interactions within lakes. Instead, we saw this seasonal reset hypothesis being supported, and so it might be that the seasonality and how the environmental change affects the onset of spring and fall turnover at the end of the season might be more important for altering species interactions than necessarily the environmental conditions within the lakes. And more generally, this use of EDM is a promising method to explore these species interactions over these really large spatial and temporal scales. We can use these types of models with any type of species interaction if you have the data. In fact, you can use these models on any time series that they don't even have to be species. And of course, we can use them in any range of ecosystems. So I think it's an interesting way to get a lot more use out of monitoring data, data that is collected with other purposes that we can now get more of these causal links out of. Okay, so for the second half of my talk, I'm going to go from this context dependence of species interactions to context dependence of stability and predictability. So first, I just define what I mean by being predictable and being stable. So if you have an ecosystem at a point, so in blue here, if you perturb it slightly from those conditions, can you predict where it ends up? So on the left, if you perturb it in different directions, you have these 3 different arrows, so they're all kind of starting slightly different from each other, but they all end up in about the same place. So that system's really predictable. Whereas in this right-hand side, you have this unpredictable system where depending on those initial conditions of these tiny little perturbations, you can end up in vastly different areas or different types of ecosystem spaces. So that system is very unpredictable, it's unstable, or another way to classify it would be it's chaotic. So it's very sensitive to these initial conditions. And so again, I guess this talk is kind of old ideas in ecology, but with new methods, because there was a discovery of chaos in ecological models in the 70s, and that kind of started this extensive search for chaos in natural systems over the next about 30 years. And so there's a lot of theoretical work on when we should see chaos, and it's been demonstrated in some experimental systems. So if you have a bucket of plankton, there's chaotic dynamics in that system where you can't make any predictions because these little stochastic deviations create these large differences in outcomes. But there's been almost no evidence for chaos in naturally occurring populations until very recently. And so why is this important? Well, there's a lot of calls for ecology to become a more predictive science. So how far out can we predict the dynamics of a system? Well, that's really going to depend on these kinds of dynamics. If you have chaotic systems, they're unpredictable, but so are systems where there's periods of chaos. So these periods of intermittent chaos where sometimes, perhaps some times of the year, these systems are stable, whereas others, they are less stable, unpredictable. And so identifying these forecast horizons, or how far out we can predict these ecological dynamics, is an important step in trying to figure out how much we can learn about our environment. So not only are we asking, are population dynamics predictable, but also when are they predictable? So are there certain environmental conditions under which systems are more predictable or less predictable? Are there certain times of the year where they're more or less predictable? And is there differences in scale? So both spatial scale, but also maybe here I'll talk a little bit about biological scale. So are they predictable at the level of population dynamics, functional group dynamics, trophic level dynamics, etc. So coming back to this method of empirical dynamic modeling, so this is what we did last time to get at species interaction strengths. So we use these time points to get locally weighted regressions to get interaction strengths. But we can also extract different types of information. So we can get something called the Jacobian and calculate the Lyapunov exponents, which are an index of stability or chaos. So a low, I'm going to call them LE, LE is indicative of something that is very predictable and stable, and something that is high is less predictable. And a system is deemed chaotic when the LE, scaled LE, is above 0. So we ran these models on the same set of lakes that I showed you last time, and here's an example of the type of results we get. So this is just one lake, and here we've got these LE plotted against the month. So from January to December, and you can see that in the winter and spring we have these chaotic dynamics, and in the summer and into fall we have more stable dynamics. So that's kind of the new part that we were able to pull out of these time series data. It's this time varying stability, so these periods of chaos and periods of stability. Previously, what we've been able to do, what people have done, is calculate the global value of LE, and that's for the whole time series. So again, like collapsing a whole time series down to one data point. And you can see here that would suggest that this system is stable and somewhat predictable. So it's only once we actually get this time varying LE values that we're able to see this chaos in the system. So this is really our first demonstration of intermittent chaos in natural systems, which is exciting. It's also what you would predict for a system to be maximally adaptable. So there's this idea of complex adaptive systems where this alternation between stability and chaos actually allows systems to be optimized. So it's called the edge of chaos. And so what does it mean that you're globally stable, but you have these periods of predictability and unpredictability? So, so it seems that there's more accurate predictions for dynamics that you're able to make in the fall, in the winter and fall, these areas of stability. And that these coarse grained predictions on the annual timescale, so that based on global stability, are also feasible, but that's mostly because of seasonal entrainment. So abundance will go up in the spring, down in the fall, though by exactly how much that would be harder to say. So one implication of this is that we might end up with a global LE, so this global stability value, around 0, because everything converges in the winter. And if winters become sufficiently mild, like this may no longer happen. So things may become less predictable if we don't have this kind of reduction in abundance and kind of resetting of the system every winter. Now, this is one species in one lake that I was talking about before, but we can look at all the different time series of the zooplankton species. And when we do that, what we find is this dark blue is the periods of time that are chaotic, and the light blue is periods of time that are not chaotic. So the species are chaotic, the species dynamics are chaotic about half of the time, but if you start aggregating those into functional groups or trophic levels, you see that those are not chaotic and they're a lot more predictable. So it's going to be a lot easier to make predictions at whole trophic level, what's the biomass of all the zooplankton, rather than individual species level. And that's true for all of the lakes, and also I believe, so there's some ocean areas that we used data from those as well. And we see that there's chaotic dynamics found in most of the species, but almost none by the time you aggregate it at the trophic level. Again, part of this analysis was motivated by climate change. So as the environment shifts, will we see increased or decreased predictability of population dynamics? So I have some predictions here. So we expected that predictability would decrease with temperature and temperature range and have this hump-shaped relationship with productivity. But I won't dwell on those because we did not find a relationship between our chaos index and any of the environmental predictors. So although we can't say anything about how predictability might change with the environment, we've definitely demonstrated that we have this intermittent chaos in nature for the first time, which is exciting. And it says something about our general ability to make predictions for these lake ecosystems and the scales and times of year at which we might be able to make accurate predictions. So overall, these new time series methods, we've used them to create these species interactions and stability metrics that are now continuous variables, so we can look at how they change through time and what they're associated with in terms of the environment or species diversity and traits. And so we can really start to use these methods to test a lot of the ideas, hypotheses put forward by experiments and theoretical models, and see if we can actually discern between these different hypotheses with data from nature. And these methods allow us to extract a lot of information from existing data sets that are just sitting there, which is excellent during COVID times especially. And this has allowed us to like I said, determine how debated concepts manifest in these natural systems. So thanks for listening. I will remind you to think about applying to Eco Days, and I look forward to talking to you about time series analysis, maybe slightly more basic time series analysis during our session together. And I'll be happy to take any questions at that time or any time in the future if you want to reach out. Thanks.